Right at 3, I'm going to go ahead and get us started. I am so thrilled to be here today for the Virology Next Frontiers in Virology webinar series from the Global Virus Network. I'm Dr. Maggie Bartlett, the program director, and we have a fascinating talk in store for us from Dr. Mark Mimee, who is a really just a fantastic voice in the space of phages and microbiota and synthetic biology. He's an assistant professor of molecular engineering at the University of Chicago and has been developing strategies to engineer the activity and composition of microbiota. And so we'll get to hear a little bit about how he does this, and in particular, how native phages can be used to selectively remove some of the resistance genes in the gut microbiome. And without further ado, I'm just so pleased that we have you here today, Mark. I'm going to hand it off to you.
Great. Thanks, Maggie, for the introduction and for the opportunity to speak here today. So I'll talk to you a little bit about sort of the phage program that we have going on in the lab, and some work that we've recently published, and also some ideas that we have going forward. But to sort of set the stage with, if I can advance the slides, with sort of, this idea of targeting bacterial infections, let's say with common antibiotics. So the overall goal really is to say you have a person that's infected with a pathogen. Here, they're sort of depicted in these hot colors. They're also kind of within this bed of commensal or symbiotic bacteria that contribute to our health. And the hope is that a patient would come in, you could treat them with a small molecule drug like antibiotics, and you would be able to eliminate these pathogens without harming sort of the innocent bystanders that would be in your microbiome. And obviously, that's not the case. These antibiotics are broad spectrum by design, so that if a patient comes to a hospital, you're able to treat them for a bacterial infection without necessarily needing to do extensive typing. But one of the downsides of that is that it also decimates sort of the commensal microbiota. And upon removal of these antibiotics, you can kind of create conditions unintentionally that would allow for the outgrowth of specific pathogens that can, in fact, worsen disease. And this is probably best typified in the case of Clostridioides difficile infections with the use of clindamycin, where this could lead to sort of these types of recurrent infections that get worse and worse over time. But you could also see a similar phenomenon as well with vancomycin-resistant enterococci, as well as Enterobacteriaceae that are resistant to a variety of different beta-lactams. So the thought is, can we, at least in some cases, move away from these conventional broad spectrum antibiotics and try to go after these narrow spectrum antimicrobials? And what we and others are interested in is leveraging bacteriophages, which are natural viruses that occur in nature. And these are viruses that can infect and kill bacteria. And the basic idea is that we can kind of leverage these natural life cycle of these viruses to be able to eliminate bacteria that are causing an infection. And kind of in this simple cartoon that was drawn by a staff scientist in my group, they kind of have the standard, life cycle of a virus, where there might be a bacterium that's living in an environment. There are viral particles that are floating around. And if there are specific proteins that are on the virus, these receptor binding proteins that bind to elements that are on the cell envelope of the bacteria, be it polysaccharides that are part of LPS, or outer membrane proteins, or pili, or things of the sort, this could allow the virus to inject its nucleic acid program into the cell. This could kind of subvert the host kind of function, allow the virus to proliferate, and this could lead to the expansion of these types of viruses. What I'll be talking about today, and what most phage research focuses on, are double-stranded DNA viruses, although there are also RNA viruses and single-stranded DNA viruses of phages as well. And kind of the benefit of these phages is that they have this narrow host range, so that they have this kind of lock and key relationship between a bacterium, so that you could use them as antibiotics that could spare the microbiome. The downside of that is that means that phage resistance is easy to evolve. Bacterial populations are large in the context of infection of multiple orders of magnitude. So it's relatively easy for a bacterium across a population level to just lose its receptor and therefore gain resistance to phage. But these are things that, folks are interested in tackling. So the basic premise of, what we wanted to do, at least at the outset, is to see whether or not we could use phages to surgically eliminate pathogens from an intact microbiome. So put it very simply, if you have a beige phage and you want to remove these sort of beige cocci from a microbiome, you could add that to a mouse or hopefully eventually a person and be able to selectively remove that bacteria in that very surgical manner. And this work was led by Ella Rotman. Like I had mentioned, she's a staff scientist in the group. It was published in Cell Host and Microbe recently. So if you want more details, you can sort of find that online. So this is the general idea, but we kind of need a model system. And the model system that we chose to pursue is going after Klebsiella pneumoniae, which is one of these members of the carbapenem-resistant Enterobacteriaceae. It's sort of the K in escape pathogens, if you've heard of this. And this is one of these highly antibiotic-resistant and sort of, high urgent public health threats to which we need to develop more antibiotics. And for a little bit of background, this is a gram-negative capsulated non-motile bacteria. And it's sort of what I call a meat and potatoes pathogen. It doesn't have any fancy virulence factors. It doesn't secrete a ton of proteases. It doesn't have crazy secretion systems or things like that. It's really a successful pathogen by the fact that it's very environmentally resistant. It could survive in a number of different habitats. It can evade the immune system very effectively. And it also happens to carry a number of antibiotic resistance genes, which make it difficult to treat. And I would say a good chunk of the folks here probably have a version of Klebsiella pneumoniae in their microbiome right now. It can colonize the gut of happy and healthy people and not cause any harm, but it's really only when it translocates to other sites where Klebsiella becomes an issue. So this could be in the bloodstream, this could be in the urinary tract, in the lung, in wounds, in the liver. It can cause a lot of different infections all over the body. And our interests, say in phage therapy, we thought that this would be a particularly good use case because it is not uncommon, say, for patients to be admitted to the hospital. Maybe they're getting ready for a transplant. They're given prophylactic antibiotics and as well as being immunosuppressed in preparation for that transplant. And if there is a multi-drug resistant strain of Klebsiella within their microbiome, that could bloom to overtake their microbiome. And then these patients could later become bacteremic with these strains of Klebsiella pneumoniae. So our use case would be, okay, well, if there is a patient that presents with Kleb in their microbiome, can we use phages to selectively eliminate it without necessarily harming all of the other microbes that confer colonization resistance? And outside of kind of the pure frank pathogen sort of characteristics of Klebsiella, also in the microbiome world, there are a number of associations of Klebsiella pneumoniae as a pathobiont, even in kind of outside of the context of infectious disease. So there's kind of two examples of that. The first of which is inflammatory bowel disease, either in Crohn's disease or in ulcerative colitis, where in multiple cohorts across the world, in Israel, in Europe, and in the United States, Klebsiella pneumoniae is consistently correlated with both Crohn's disease and ulcerative colitis, also in the context of flares. Meaning that, in these, and these are also specific clades of Klebsiella. So in these patients that have a complex microbiome, again, you might not want to treat it with antibiotics, but you might want to use phages to kind of selectively remove the Klebsiella from there. This is also the case in fatty liver disease, where there are some patients that have non-alcoholic fatty liver disease that have elevated levels of Klebsiella within the microbiome. And again, these are specific subtypes of Klebsiella that quite interestingly produce a lot of ethanol as a primary fermentation product. So even though these are non-alcoholic versions of fatty liver disease, the sort of alcohol that's being produced endogenously by the microbiome kind of induces this constitutive stress on the liver that can exacerbate disease. Anyways, so the general goal of what we wanted to do is kind of create a bank of phages that we could use to be able to target multi-drug resistant Klebsiella because we think it's an interesting pathobiont or frank pathogen. So what essentially I'll do for the remainder of the talk is sort of walk through this little pipeline that we had, which is building up banks of Klebsiella and phages, choosing which ones we should combine within cocktails, sometimes trying to evolve individual phages so that they can optimize their virulence on specific Klebsiella hosts, and then later use these optimized cocktails within the context of a mouse to see whether or not they can cure an infection. So the first thing that we needed to do was isolate phages. So we did this in very conventional manner where we had Klebsiella strains that we would collect from a variety of different sources. We would get source material, which for us was going to a local water reclamation plant, asking for gallons of sewage, getting that sewage. And what you do is mix together the bacteria with the sewage and then pour it on plates. You'd see these zones of clearing that you call plaques. You get happy if you get some of these. You streak them out a couple of times, you amplify them, and then you have these virus particles that you can infect. So with that, we were able to build large collections of these different phages on many different types of Klebsiella. So I'll walk through this a little bit. So kind of the cast of characters on the bacterial side of things are mostly strains of Klebsiella pneumoniae and quasi-pneumoniae. We have some varicole and oxytoca strains as well. They kind of take two different flavors. So the first of which are carbapenem-resistant strains, or these highly multi-drug resistant strains that we kind of prioritize targeting. And the second of which were primary isolates that we were getting from the urogynecological clinic from patients that had recurrent urinary tract infections. And this is with the hope that eventually we would be able to use these phages in that particular patient population. And Klebsiella, without going into too many of the different details, across our collection of Klebsiella, we have many different types of capsule or O antigen types. Capsule is sort of one of the major factors in kind of phage bacterial interactions with Klebsiella, because it's really the outermost surface of the bacteria. So this is the first thing that a phage would see when it's trying to infect. So some of our phages are capsule dependent, and we have a wide range of capsules so that we can kind of capture that capsule dependence when necessary. So those are the rows. Along the columns are a variety of different phages that we have. So there's TEM images of sort of our representatives from the major taxa up top. The nice thing about also working with Klebsiella is that the Klebsiella phages also have many different similarities to E. coli phages, which are obviously kind of the most studied phages that exist. So we're allowed to sort of borrow a lot of the findings that exist in E. coli phages and kind of port them directly onto Klebsiella phages. In this panel that you see here, the degree of shading indicates kind of the degree of killing. So the particularly dark colors are very effective phages, whereas the lighter colors kind of get very minor clearing. And you can see kind of just looking at the different, they're also color coded according to taxa. And you can see according to that, there are definitely some taxa that seem to be broader host range, like these T4s, the blues and the red phages, as well as the purple ones that we see here. But then there are also others that are in the negative space that are much more narrow host range that are only infecting very specific types of Klebsiella. One of the fun things that we found in kind of amassing this collection in Chicago is that for whatever reason, our Chicago sewage is very enriched with these purple viruses that we've, named a new genus called Purpuraviridae. There only a couple of these representatives were kind of previously published or available in databases, but for whatever reason in Chicago wastewater, we have a ton of these viruses that can infect these multi-drug resistant strains of Klebsiella. And this is really just kind of also a snapshot of the phage collection, at the time of, well, at least when we submitted the publication. Or here, there's around 50 or so phages. Now we have over 100 in the lab, and we're also part of a bigger consortium that's trying to grow this to 1,000, so that we really have this kind of massive collection, specifically for these narrow host range phages that we could use to cobble together to put into cocktails. One other thing that I usually get questions asked about, so I'll preempt this here, is we did some analysis as well of the Klebsiella strains to look for defense systems that would be present in the bacterial genomes to see whether or not that would predict kind of overall levels of susceptibility or resistance to the phages. So the bacterial strains here are rank ordered according to susceptibility, where the most susceptible strains, i.e. the ones that are most infectable by phages, are up top, and the ones that are least infectable are the ones that are at the bottom. And we could see kind of this loose correlation between the number of defense systems that would be present in the bacterial genome. So these could be things like restriction modification systems, a variety of different CRISPR systems, or retrons, or even these newer ones that are continually being discovered. And we could see that by and large, the more of these defense systems that a strain has, the more resistant it is to any sort of random phage. But we could also see, too, that if we kind of put in the effort to isolate phages against specific strains, that even though that there are a number of defense systems, we can get kind of more highly resistant. We could still find, highly virulent phages that can clear these bacteria. We also looked at the number of prophages in the genome, because there's some reports that prophages can kind of exclude infection from other phages. And there we see a very weak to non-existent correlation, kind of pointing to the fact that maybe these kind of more sophisticated defense systems would be more important there. Obviously, a ton more work to go into which ones are important and which ones aren't super important. Cool. So with this, we had kind of our huge bank of phages, and then we get to the question of how do you go about selecting what the optimal phages are for a cocktail? And there, the kind of simple premise is that we will want functionally distinct phages. In order phages that if a bacterium were to evolve resistance against one phage, it wouldn't evolve resistance to the other phages that are in the cocktail. So that sort of sent us down a rabbit hole to try to identify what host factors are required for phage infection so that we can map out sort of how bacteria are becoming resistant so that we can design how to put together these cocktails. And to do so, we use kind of a TN-seq-based approach where we had a library of Klebsiella, and this is our favorite strain. It's this Klebsiella pneumonia strain MH258, which is one of these carbapenem-resistant strains. It's a clinical isolate from the bloodstream of the patient. And we kind of inserted transposons, or rather, we got a library in which transposons were randomly inserted across the genome of the Klebsiella. So each individual cell, or there would be subpopulations of cells, would have a knockout in a given gene within its genome. And then we can challenge that population with phage. The resistant bacteria, or bacteria that are resistant to the phage, would expand. Those that were still susceptible to the phage would be killed by it. And there we can kind of map where those transposons are in the genome, either just by plating colonies and doing arbitrary PCR to find where the transposon is, or using Illumina-based technology and mapping where those transposons are in the genome. From there, we got a list, and for the candidates that we thought were the most interesting, we did make clean knockouts using lambda-bred recombinering and complemented those on plasmids to sort of convince ourselves that what we saw in kind of these pooled screens really is an important host factor for phage infection. And we did this with 5 taxonomically distinct phages in our collection, the details of which aren't necessarily all that important. And we found that, the majority of the hits that we would see are different components of the cell envelope. So these could be things like capsule, these could be things like LPS, either specific modifications that are made to the LPS or whole elements of the O antigen. You can also see, too, that some of our phages are capsule dependent, like this Phi-ER12, as well as this ER46, where the capsule is the major receptor for these phages. Other ones are LPS dependent, and yet others are dependent on outer membrane proteins that are on the surface of the bacteria. You can also see that there are multiple paths that a bacteria could take to become resistant to a phage. So for example, for this ER39 that's here, either a mutation in a specific LPS modification enzyme WAH, or the loss of OMP-C, which is an outer membrane porin on the outer membrane, that both of those could lead to resistance to a particular to this particular phage. On the bottom, there were some of kind of the weirder things that we've seen. So for the first of which is this mutation in GAL-U, which is a very upstream component in both the biosynthesis of lipopolysaccharide as well as the capsule. This was the only mutation that we saw that can confer resistance to all 5 of these phages that we saw here. But interestingly, this was only in liquid culture. If you would spot on plates, like you can see with ER46, that strain was still able to infect, but the strain was resistant in liquid culture. So that might be kind of a downside of, of maybe using these phages, because there is kind of a solution that the bacteria can find. The good thing is that these GAL-U mutants are terribly sick. If you would put that in the context of a microbial community, they would die almost immediately. So this is really just sort of an artifact of being able to challenge these in the lab, but you would never see this in the context of infection. The other interesting thing that we saw was a mutation in this transcriptional regulator called CSRD, which is a component of this broader regulatory system that regulates a transcription factor called CSRA, which stands for carbon storage regulator. So this has been studied extensively in E. coli as an important factor that bacteria can use to shift their metabolic strategies from one sugar to another. So we were a bit puzzled as to why this would be related to phage infection. So what we did is we did transcriptomic analysis of both our knockout as well as the overexpression mutant. And as you can imagine, it being implicated in a sort of regulatory pathway, we saw many genes downregulated as well as many genes upregulated. Some of these were involved in the synthesis of peptidoglycan or other outer membrane components. We also saw downregulation in the mutant of genes involved in purine metabolism, and nucleic acid availability is extremely important for a phage to get in and be able to infect effectively, as well as things that are involved in starch and sucrose metabolism that largely correlated, again, to sort of synthesis of LPS or capsule. So if you look at the spots that are on the right, you can see that it doesn't confer kind of this very stark resistance phenotype like some of the other knockouts, but rather makes the strain less susceptible. So So we think that the mutation in this kind of transcriptional regulator is kind of changing the phenotypic state of the bacteria to one that's just less conducive to viral infection. So that's fun. So the last bit of this is seeing whether or not we can evolve specific phages to be more efficacious at clearing bacterial populations. And the basic rationale behind this is that we were effectively, taking random strains of Klebsiella that other than the ones that we were getting from the clinic, were kind of from a variety of different culture repositories from around the United States. And we were taking Chicago phages, so these random ones, isolating them on a Klebsiella strain, and then trying to see whether or not they would infect different bacterial hosts. But it wasn't necessarily the case that that phage stock that we had was kind of the optimal phage to infect that specific Klebsiella strain. So what we would do is we would take each individual phage and then passage them on our target strain continually passage it on the ancestral strain so the bacteria don't get a chance to evolve. And then at least for some of the phages, what we would see is that there would be mutations that could be sort of fixed within the phage populations that would allow them to infect these populations better. So there's kind of two examples of this that we saw. So the first is in this T4-like phage of ER16, where if we did this sequential passaging, we saw this point mutation in its tail fiber, so in its receptor binding protein, that would allow better absorption of the virus to the bacterial cell, as well as more effective killing when you compare it an equivalent MOI. We saw we saw a similar thing with another phage called ER46, and this was even a more stark phenotype that we kind of stumbled on by accident. So this is the wild-type phage, wild-type bacterial strain, MH258, and its susceptibility to a variety of different phages here. And for ER46, you can kind of see this very faint clearing, but it doesn't infect very well at all. Somewhat fortuitously, while we were kind of doing sort of these assays and trying to find out knockouts, we found that one of our overexpression strains, when we were trying to complement the CSRD mutant, when we would overexpress this CSRD, it would actually sensitize the strain to a variety of different viruses, including ER46. So we figured, okay, maybe we could use this as a host of MH258 to sequentially passage ER46 since it's more susceptible, and maybe this would give ER46 a leg up so that it can infect the wild type host. And that's exactly what we saw. We saw that it got this very large 400 amino acid deletion in the main receptor binding protein in ER46. And this could render the phage that could barely infect a wild type strain of bacteria into one that was one of the best clearers that we had. And again, we can map this to absorption, that the deletion of this large domain within ER46 allowed the phage to bind to the bacteria a lot better, and this would allow it to be a significantly better phage. Cool. So with these, with this kind of information in hand of what are the host factors, as well as some of these involved mutants, what we're able to do is to be able to put together different combinations of phages that can minimize the evolution of resistance. So again, this is for MH258. These are 5 different phages that are all taxonomically distinct and dependent on different host factors for infection. And you can see up top with the individual phages that we can get clearance of bacterial cultures. But if you look on the bottom, if you take this time course out over time, you can see that fairly reliably with the individual phages, you can get the evolution of resistance at a fairly distinct and specified times where you select for these bacterial mutant populations. But when you combine them all together in a cocktail, which is in the teal line, you get very few, if no, populations that can evolve resistance there. And since we have these collections of phages, we're able to rapidly do this with different strains of Klebsiella. So in the middle column is a Klebsiella quasi-pneumoniae strain that we got from our clinic from a recurrent UTI patient. And there we can cobble together a couple different phages, very effectively suppress resistance. And the same with another Klebsiella strain on the right, which is one of these extended spectrum beta-lactamase strains. So with these, we wanted to say, okay, we had these, cocktails of phages. Can we use them to sort of go into a mouse that has a complex microbial community and see whether or not we could remodel it? And this is where, things get a little bit hairier and a little bit more difficult. So to set it up, the model that we're using here is a gnotobiotic model of mice that has a defined community of bacteria. So this is something that we use very frequently in the lab, a little bit for the phage work that we do, but much more so for the microbiome work, where we can use this defined microbial community, where we know all of the functions of the different members of the microbiome so that we can get down a little bit deeper into mechanism and also sort of, not necessarily have the variability, mouse-to-mouse variability in working with native microbiomes. So what we're able to do with this model is we have this community of 13 bacteria that we house in an isolator in perpetuity, and we can just give Klebsiella to these mice. And since there's not necessarily full colonization resistance there, we can get Klebsiella to engraft within the microbiome somewhere between 0.1 to 1%. And we also like this model as well because a lot of other SPF models that use mice often perturb the microbiome using antibiotics, and Klebsiella then becomes, 100% of the microbiome. Here, we're able to sort of create a state where Klebsiella can exist in the microbiome at these low levels, so we can see whether or not the phages can kind of remove them from this community. So what we did is we had, we had these mice colonized with the different bacteria. We would treat them both with phages, and we got somewhat disappointing results. And seen with two phages, two administrations of phages, we could see a drop in Klebsiella titer, but it's really just a half log and it's quite variable. So we looked a little bit more into it, and we could see that part of this is just due to delivery of the phage to the distal gut where the Klebsiella are living, where in the mice where we're able to get much higher titers of phage into the distal gut, we can get much starker sort of decreases in Klebsiella. But in many of the mice where the phages are clearly degraded as they're making their way down the gastrointestinal tract, we don't get all that much clearance that's there. So this is pointing to the fact that both we or maybe others in the field need to think about different sort of formulations to improve the bioavailability of phages, especially for these types of gut applications. And so even though this was kind of, mediocre data, there's kind of some interesting things that we learned from it that are kind of fueling sort of future research. So the first of which is this thing that we've observed and what others have observed as well, is where you can see this coexistence of virulent phage and susceptible bacteria within the same mouse. And by that, I mean, if you take a mouse that you've administered phage to and you try to isolate Klebsiella colonies, they're still susceptible to all the phages in the population. And likewise, if you take the phages in the population and try to infect the original colonies, that you could still see that they're infectable. So this is pointing to the fact that there's definitely factors that could largely be chalked up to heterogeneity that exist within the mouse that are allowing for the coexistence of these phages and bacteria to occur at the same time. We also saw as well is that there are relative changes in virulence of specific phages when you take it from the test tube setting and move it into the gut. So on the right are these plate reader curves that I had shown before with the 5 different phages that are in the cocktail. And if you kind of rank order which phages are best to worst, it's quite interesting that actually our best in vitro phage doesn't infect at all in vivo. This orange phage is just rapidly cleared, and that's just it transiting through the gut. Whereas our worst in vitro phage, this purple phage here, which is barely even kills cultures in vitro, is by and large the most successful gut phage. So for us, this is pointing to the fact that we really have kind of a poor understanding of how the environment really impacts the success or failures of putting together phage cocktails, and also suggests too that maybe all that sort of prototyping work that we were doing up front in vitro is probably not being done in the right setting, and a lot more work needs to be done to sort of defining what a better model system would be to actually understand how bacteria and phages are interacting in the context of infection. But what I will say, all is not lost. What we did notice in kind of doing these experiments is that upon administration of the phage, we can see these visual shifts in the populations of bacteria that we were able to recover from the mice. So at first we were worried because we thought that the E. coli that was in our microbiome was maybe sneaking through onto our antibiotic plates. But in fact, we found that there are the capsulated strains of Klebsiella, that we had introduced into the mice and that were colonized. Once we applied the phages to them, we would see that these populations of Klebsiella would lose their capsule, which is actually their major virulence factor. So we followed up on this to see whether or not this would be important. And we did kind of this macrophage phagocytosis assay to say, okay, if we use phages and we treat bacterial populations with them, can we sensitize bacterial populations to elimination by the host immune system? So this is some in vitro work that we did where we had Klebsiella pneumoniae. We would infect them with a phage population. We would take their survivors and we would effectively feed them to macrophages. We would see how many of them would get phagocytosed, treat them with gentamicin to kill extracellular bacteria, and then see how many were internalized by the macrophages. So here's kind of a series of both knockouts that are denoted by the deltas, as well as phage-treated populations by individual phages. And beneath, I'm letting you know if these are capsule-dependent phages or LPS-dependent phages or these sorts of things. So as kind of a control, we know it's known in the literature that acapsular bacteria are more readily phagocytosed by macrophages. And we can see this, that when normalized to wild type, we can get kind of a 200% increase in the amount of phagocytosis for an acapsular bacteria. And what also makes sense is if we use a capsule-dependent phage, so the majority of the bacterial populations therefore become acapsular, we can again sort of increase the sensitivity of these bacterial populations to macrophage phagocytosis. And we did this with a variety of different mutants, and we see that LPS mutants, but not outer membrane protein mutants, are important for sort of sensitizing these populations to phagocytosis. What's interesting, though, is literally every single phage that we use, even those that don't necessarily depend on capsule or those that depend on LPS, we can still see the sensitization phenotype, sort of implying that maybe some factors of the phage infection just at all and the kind of bacterial survivors render these bacterial populations more sensitive to host immune clearance. So with that kind of information that maybe there's not this one-to-one sort of relationship between capsule loss or susceptibility to immune clearance to the phages that were used, we repeated this kind of capsule loss assay with a larger panel of phages. So on the left is kind of the no phage control, where there's very few of the recovered mutants become acapsular. In the second column is the 5-phage cocktail that has all the phages there. So that's the same data that I showed before. And then we chose a couple of different subsets. So the first of which is this ER12 subset. This is a capsule-dependent phage by itself. And you can see that there's this massive variability in some cases, we see some populations, a lot of it is lost. Others seems to do nothing at all. And what was quite interesting is this middle one that has the yellow, the red, and the orange phage. 2 of these phages are capsule dependent. And there we see that there's very little sort of capsule loss there. Whereas the best population that we saw were this blue, red, and this and orange, where none of the populations, none of these phages are actually dependent on capsule, but we can still see this massive loss in capsule. So this points to the fact that we don't necessarily understand how bacteria are kind of responding to phages, how they're evolving around phage infection within the context of infection. And sometimes you can see these kind of unanticipated results. When we saw this, we kind of looked more deeply. Is there more than capsule that we're seeing within these populations? And in fact, that's the case where we can see multiple different populations that emerge on the bacterial side when we challenge the population with phages. We see capsule loss that's reflected by this WZC mutant, but we also see things like loss of O antigen in the WBBO, the WBBN, and the WZM mutants, as well as mutations in LPS with WAH and WALW. And on the right, this is the data from the macrophage sensitization assay. We see that some, but not all, namely the capsule and O antigen mutants, allow sensitization to macrophage phagocytosis, while some of the other kind of LPS mutants don't necessarily have an effect. Cool. So we thought that was fun, but then we decided we were focusing way too much on the bacteria here. Let's get back to the phages and see what the phages are doing. Because we figured that if bacteria were responding to phage challenge by evolving resistance around it, maybe the phages were evolving ways to counteract those mechanisms of resistance. So the first thing that we did was just take fecal contents from these infected mice and do sort of whole genome shotgun sequencing to see whether or not we can detect variants within phage populations at a population level that were occurring after we were treating these mice with these. We only got the read depth for phi-ER39, which is the one that was at the highest titer within the gut. This is one of these purple phages. It's a broad host range cyphovirus, and we thought it was also perhaps interesting as well, because this is definitely an understudied virus. And what we can see is that across the genome, we can see diversification of the ER39 population, with many of these resistant mutants occurring within structural proteins like the tail fiber. That points to the fact that there might be sort of, again, mutations that are happening that allow these phages to bind to different sort of receptors on the bacteria in the context of infection. So with these phages, what we decided to do is isolate individual plaques from these populations, sequence their genomes, and see whether or not there was differential infectability with these different mutants. There's a lot of data on this slide, but the point that I want to get across is that we're definitely able to see mutations that tend to fix in 2 specific regions within the phage genome. So the first of which is a hypothetical protein of absolutely unknown function and no structural homology. So there might be some interesting biology there, but not not much to learn at first glance. And the second of which are there's mutations in the tail fiber of the protein, sometimes in different sugar binding domains that are annotated, or sometimes immediately adjacent to it. It's also interesting too that if you look at the size of the genome, this lambda-like tail fiber takes up almost a quarter of this phage's genome and has multiple different sugar binding domains. So it might suggest the reason why this small phage, which normally has a very narrow host range, is actually able to infect a variety of different phages because it has this kind of multifaceted kind of Swiss Army knife receptor binding protein. Anyways, one of the main things that we saw is that these phage mutants that we're able to isolate from the mice with these mutations in their tail fiber, they're able to infect this WBBO mutant of Klebsiella, which is an O antigen mutant that we would see pop up kind of in the bacterial populations very frequently in the mice. With these mutant variants as compared to the wild type, we can see that they're able to bind a lot better and that they have very similar EOPs compared to that. So with that together, we kind of have a model of what's happening with ER39 in the gut, where normally you have a Klebsiella population in the mouse gut that's just, living along. And if you introduce ER39, it's able to infect using the O antigen as a co-receptor. The bacterial population responds by losing that O antigen, so that now resists the ER39 infection. But now certain subpopulations of ER39 can mutate the receptor binding protein to then infect that O antigen mutant and sort of maintain high levels of the phage within the gut over time. Cool. So with that, what I hope you can appreciate from what I showed today is that these phages can, to some extent, suppress the burden, but also suppress the virulence of Klebsiella pneumoniae within the gut microbiome. And while this was, somewhat negative data, at least for me, this is very exciting because it points to the fact that these in vitro predictions that we were making don't necessarily match in vivo efficacy. So for, grad students and postdocs that are interested in working on this in the group, we now have a number of sort of avenues of things that we want to go after. So the first of which is, maybe a little philosophical, but getting after this question of what is a gut phage, meaning are there specific taxa of phages that are particularly adept to infecting Klebsiella in a gut environment versus Klebsiella in the soil versus Klebsiella in urine? Are there taxonomic determinants of that, or are there genetic determinants of that? And can we sort of at an omics level be able to predict if given a given phage, which one would be best suited for a particular environment? The second thing that we're interested in is trying to better define the phenotypic state of gut-resident Klebsiella. So it's quite obvious that, depending on the gene expression profiles of a Klebsiella strain, that might affect its susceptibility to a phage. For instance, if it's expressing a couple of different receptors, if it enters the gut and then downregulates all those genes that encode for those receptors, that might allow that population to be more resistant. So we're trying to sort of figure out whether or not that's the case. Is that the case for all phages? Is that what's governing what's a good gut phage or a bad gut phage, and all those sorts of things? We're also trying to look into as well whether or not the spatial niches within the gut kind of confer some sort of protected nature of Klebsiella. For instance, maybe the Klebsiella are hiding away in the mucus, and all these phages that we're using can penetrate the mucus layer very effectively. So can we use that kind of spatial niches to infer what's going on? And with all those sorts of things, what we're interested in doing, is seeing whether or not we can come up with engineering strategies to get around this, either selecting for better phages, designing better model systems, engineering phages so that they have better mucus diffusivity, all those sorts of things. I had alluded to this earlier, but now this work is part of a larger phage foundry, which is out of the Lawrence Berkeley National Lab, where there's a number of us that are trying to build these repositories of phages so that we can systematically design phage cocktails for multi-drug resistant pathogens. We're also interested in using kind of a variety of different AI and ML approaches to see whether or not, just given a bacterial genome and a phage genome, whether or not we're able to predict infectability, and also whether or not be able to predict synergy between phages and or antibiotics when combined within cocktails. I had mentioned that we're doing some engineering work as well, and what we're also hoping to do is take some of these phages and some of these applications that we have and bring them to the clinic with some medical doctors here that are quite excited about the prospect of this. The first thing that we're interested in is for recurrent urinary tract infections, particularly in elderly women, where antibiotics, don't seem to be working anymore. So the idea is, can we use phages to both treat the microbiome of these women as well as their bladders to see whether or not we can cure these recurrent infections? Cool. And with that, I'll acknowledge just sort of the lab and everyone that worked on this. Like I mentioned before, Ella was sort of the lead author in all this work, but more or less everyone in the lab contributed in one way or another, whether it was for mouse sacs or cloning or doing macrophage assays or that sort of thing. So I'd be happy to take any questions now. And if anyone is sort of interested online and too shy to ask a question, you can feel free to shoot me an email. Thanks.
Thank you so much, Mark. That was a fascinating talk and just truly a tremendous amount of work to try to parse these complex systems biology level interactions. And I wonder if in the time scale of this being a therapeutic approach, where do you see that? How early are we? And is it 5 years out, 10 years out? Um,
It,
what's the trajectory looking like?
so I guess it depends. So I think phage therapy has strong legs for very certain types of infections, particularly chronic infections that are difficult to treat, and that you have the time to design a bespoke phage cocktail to treat that particular patient. At least in the United States, from a health economic standpoint, that's a little bit of an issue because being able to say have a patient that's admitted to a random hospital, and then you would need to ship out a bacterial samples so that some company or some government agency would be able to design an optimal cocktail that is only specifically used for that patient, and then that could be sent back and to treat that patient. That is quite difficult, at least with the current structure of our economics. There are definitely some smaller countries in Europe, like Belgium, as well as Israel, has a fairly robust phage program where there are these government-funded agencies where folks with these chronic infections, even across Europe, will go to Belgium so that they can get this type of bespoke specialized therapy. But whether or not that is, possible in a much larger country like the United States, that kind of remains to be seen. So I'd say from a scientific standpoint, there is still a lot to learn about, phages and when they're appropriate and that sort of thing. But also from an economics and kind of application-based standpoint, I think that's maybe the bigger challenge for phage therapy than does it work or not.
Interesting. And Sten asked a question about safety and efficacy. What's the status on randomized control trials currently? I know you're setting one up for, which I think will be fascinating for the urinary infections, but is it similar where it's very different in the US to other parts of the world?
Yeah. So I mean, there's like, I guess we work on in the lab, but I didn't talk about it today. So again, one, you're getting into the randomized controlled trial. So there's a couple of them that have been done in the past. They've been abject failures for one of two reasons. One, because very little effort was paid towards the delivery of the phages. And in fact, in one of the trials, they thought they were delivering whatever, 10 to the 8 PFU per patient, and they didn't titer it adequately, and it was actually 10 to the 5 PFU. So it's no wonder that it didn't work because they actually weren't delivering the drug. And then the other ones too were the ones that failed, where they were hoping to kind of create this master phage cocktail that had 12 different phages that was able to cover every Klebsiella, or sorry, Pseudomonas that would come into the hospital. But it turned out that that wasn't necessarily the best approach. It's very difficult to find patients that were actually had infections that were susceptible to their phage cocktail, so that clinical trial ended up failing. What's going on now more in the United States is biotech companies that are picking up the kind of phage garb and trying to see whether or not this could be applied. So there's a company called Locus Biosciences, which is going into phase 2 trials now for a variety of different infections, mostly E. coli's and things like that. They have a phage that's equipped with a CRISPR, so that along with the natural infection of the phage, it's able to kind of shear the bacterial genome, and they see that it's much more efficacious at clearing infections. And that kind of element of phage engineering allows the drug to be patentable, which isn't necessarily true with phage cocktails. And that also allows the sort of biotech to kind of buy in and say that there's actually a value proposition here. Say if we were to take our natural phages and say we're going to deploy them, we wouldn't have any intellectual property behind that. So it'd be very difficult to raise enough investment to actually fund a randomized controlled trial at the scale of which you'd be able to get FDA-approved therapy. So for a lot of these natural phages right now, they're mostly being used for compassionate use cases, again, for these patients of which there's no kind of treatment options available. And there's kind of a one-off, we'll design a cocktail, we'll use it to treat this specific patient, we'll see whether or not it works. And if you look at those series of case studies, you can see massive success in the United States, in Europe, in Israel, kind of across the world. But whether or not that's kind of scalable to the size of a country, that kind of remains to be seen.
No, that's fascinating. And I see from Chadwick, thank you for this in-depth presentation. In the long term, what would the cost-benefit comparison be like for phage-based therapeutics compared to traditional antimicrobial approaches, particularly for recurrent UTIs, in developing nations or resource-limited settings?
Yeah, well, that's a good question. To be honest, I don't really know. So I'd say compared to at least some conventional um, antibiotics like that would be shelf stable, phages aren't necessarily shelf stable. So when you're going to resource limited conditions, that would definitely be a challenge in making sure that your phages are viable there. And like I mentioned, it's like that I think they'd really only be useful for specific cases in which antibiotics are failing. If a patient, you said recurrent UTIs, but if a patient comes into the hospital, in an ER and they're expected to be septic, you wouldn't want to risk using a phage. You would want to take every single antibiotic that you have and completely blow out the infection, because that is kind of the urgency of what's had. So it would really be for specific use cases. And I would also say too, in resource limiting settings, this really, I think phages would work probably the best when they are designed custom based for each patient. And by definition, that's very expensive. So in some resource limiting settings, that might be quite difficult to at least produce, in sort of FDA compliant GMP type manner.
Sure. And I wonder if you might comment on how artificial intelligence is maybe being used in this space, whether that's predicting between in vitro and in vivo, or I'm sure in the um, what's most helpful on the synthetic biology side, but I
Yeah.
imagine that's quite a hot topic too.
Yeah. So there's definitely a lot of folks interested in that. So I had mentioned that we're part of this collaboration with Lawrence Berkeley National Lab, and there's some folks there that are really focused on that. I'd say like the state of the art right now is hoping to be able to do a fairly accurate genotype to phenotype prediction, saying like, okay, we have a phage, which strains might it infect from a genotypic level? And you'd think that might be easy, but it's actually very non-trivial. Just if you gave a phage expert a random phage and say, like, who do you think this will infect? Maybe they can tell you at a species level, that's easy, but when you're getting down to a strain level, that can be quite difficult. So folks are looking at it there. And I also mentioned too that I think there's a lot of work that's kind of being done across biology. If I can go back, can I go back? So for example, like looking at this Phi 39 genome, the gray proteins are those that are labeled as hypothetical proteins. So you can see pretty much 50%. This actually genome is actually not that bad, but I'd say in general, about 50% of a phage's genome will be annotated as hypothetical proteins. Oftentimes there are these clusters of proteins that are near the end of the genome. They're dispensable under lab culture, but who knows when they're necessary. You could see for a lot of the work that's going into phage defense, a lot of these proteins tend to be sort of these conditionally expressed proteins that are useful in kind of subverting the bacterial immune systems, or, helping in other conditions and things like that. But I think kind of parsing out what the function of these hypothetical proteins are is going to be another application of artificial intelligence, as you're kind of being able to structurally predict what these proteins might be and kind of map those across larger databases and try to infer their function there. I think that can kind of very rapidly kind of unfurl a lot of these mysteries around phages and what these proteins might be.
Yeah, such an excellent point, and underscores the importance of molecular virology and microbiology and being able, we don't know 50% of the proteins in this specific phage. There's still a lot to discover. So
Yeah,
um,
yeah,
I don't see any other questions in the chat. I want to thank you, Mark, so much for a fantastic presentation, and for joining us today, and for everybody who's on the line today. Next month, I hope you join us to hear from Dr. Patrick Dacombe, who will be talking about HIV and TB. And without more to say, I'll end the webinar. And thank you again, Mark, so much. It was fantastic.
Great. Thanks. Thanks, Maggie. And thanks, everyone. See ya.